Hi all, in this video we're going to be looking at mitosis and meiosis. First of all, you need to know that both of these are examples of cell division, so how cells divide and multiply. Mitosis is a type of cell division that produces two daughter cells that are identical to parent cells, so they have the same number of chromosomes, which will be 46. The uses of this is to create body cells for growth and repair. Whereas meiosis is a type of cell division that produces 4 daughter cells that have half the number of chromosomes as the parent cells. Uses of this is to production of gametes, or sex cells as they could be called, which is examples are sperm and egg cells in humans and spores in plants. So the key thing that you need to know here is meiosis is identical cells, same number of chromosomes, which is 46 in the new cells that are formed, whereas meiosis, they're different number of chromosomes to the parent cells. They're actually half, which is 23 chromosomes. So a little recap first. Here we have a picture of a cell with a cell membrane, a nucleus, nuclear membrane, and chromosomes. A reminder that there's 46 chromosomes altogether, however, I've only 4 shown here in the diagram, or 2 pairs. If there's 46 chromosomes altogether, that means there's 23 pairs in one cell. And a reminder that a gene codes for a particular feature or characteristic. So for example, eye colour, hair colour, genes code for everything in the body. And if they're found in a pair, so you have 2N, if we represent one chromosome as N and the second chromosome as another N, so the two of them together is 2N, we would call them diploid. So it's just a shorthand way of writing diploid. First of all, mitosis. There are several stages to mitosis, and the first is interphase. So here again, I've just made an easier diagram for this one. You can see that the nucleus and the cell membrane. Interphase is the longest stage of the cell cycle, and that's because this is where it spends most of its life, will be in interphase. However, in preparation for mitosis, the cell copies DNA during interphase, so it replicates the DNA. So what you end up getting here is twice the number of chromosomes. So if there's 46 chromosomes in a normal cell, multiply that by 2, and you'll get 92 chromosomes during replication. You can see that the chromosomes are copied. So I've indicated that by a light colour blue and light colour pink. And the copied chromosomes remain attached to their original counterparts. So you can see I have the original ones in dark red and dark blue, and the copied ones are attached to them. And where they're attached, I've just put a black dot on them there, is what we call the centromere. So that's the point of attachment, the centromere. During this time, ribosomes and mitochondria are also replicated. This brings us to stage 2, which is prophase. So during stage 2, the nuclear membrane disintegrates or breaks down, or as we can say, disappears. So you can just remove that. The centrioles are formed in opposite poles. So I put these as green dots. So these are centrioles. And centrioles have fibers attached to them, called spindle fibers. Okay? So these are your spindle fibers. Your chromosomes are now free to move around the cell because they're not contained in the cell or the nuclear membrane anymore. So the next stage is metaphase. So here, double chromosomes line up in the center of the cell. So you can see now they're no longer wandering around, they're in a distinct line. The spindle fibers attach to the centromere. And the next stage is anaphase. So here, the centromere splits, okay? And the chromosomes doubles are separated. So the original and their copies are separated and are dragged to the centrioles. Okay, so 46 will be going in one direction, 46 will be going in another. So half the chromosomes move to one pole and half move to the other. You will also notice during this time that the cell membrane at this stage starts to pinch. So what I mean by pinch, it starts to dip in the center. This then brings us to the last stage, which is telophase. And telophase is when the cells start to separate. So cell membranes begin to divide. Nuclear membranes begin to form around the chromosomes until two identical daughter cells are formed. These have the same structures and number of chromosomes as the parent cells. They are also diploid, so they'll both have 46 chromosomes. Now, when it comes to meiosis, a lot of the same things happen again. So we start with a parent cell. This time with 46 chromosomes, it is diploid. The DNA is replicated, so we're multiplied by 2, so we have 46 chromosomes. That splits into two daughter cells again, which are both have 46. Now, this is where it changes. For meiosis, the daughter cells divide again. Alright? To form the true daughter cells. Okay, so these 46 chromosomes divide again. That is not the end of the stage. Okay, and what you'll notice is that you'll get 4 new daughter cells, and the chromosomes aren't found in pairs. So previously I had a dark and a blue chromosomes tucked together, or red and pink chromosome. You'll see now that we have red and blue, but these aren't found in a pair. They're found by themselves. Okay, that means they have half the number of chromosomes. So each one of these new daughter cells have only 23 chromosomes. And these will be examples of sex cells or gametes. Now, what is the point of having 23 chromosomes being produced in mitosis? It is not enough to form a true cell. Well, during fertilization, you'll have a sperm and an egg cell meet. And 23 and 23 will join together to form 46 chromosomes, enough to produce a brand new cell. Hopefully this makes some sense to you.